Hello guys, welcome to my YouTube channel. So today we are going to discuss tryptophan operon. Now tryptophan operon is an operon that contains a set of co-transcribed structural genes which code for intermediate enzymes involved in tryptophan biosynthesis. So since this operon is involved in the biosynthesis of the amino acid tryptophan, so this operon is also called as an amino acid synthesizing operon. So this tryptophan operon is sometimes designated as trypoperon. So now let us see the structure of the tryptophan operon. This tryptophan operon has 5 sets of structural genes designated from A to E. These are tryp A, tryp B, tryp C, tryp D and tryp E. Alright, now these 5 sets of structural genes are co-transcribed together, and these 5 structural genes on transcription and translation produce some intermediate enzymes involved in the biosynthesis of the amino acid tryptophan. If you see, the tryp E structural gene codes for the intermediate anthralate synthase component 1. Likewise, tryp D structural gene codes for the intermediate anthralate synthase component 2. Tryp C structural gene code for the intermediate N5'-phosphoribosyl anthralate isomerase indole 3 glycerol phosphate synthase. Tryp B structural gene codes for tryptophan synthase beta subunit, and tryp A structural gene codes for tryptophan synthase alpha subunit. All these intermediate enzymes are involved in the synthesis of tryptophan from its precursor chorismate. If you look carefully at the bottom, the detailed process of synthesis of the amino acid tryptophan from chorismate has been shown utilizing these intermediates which are produced from the 5 structural genes present in tryptophan operon. Now, in tryptophan operon, apart from these 5 structural genes, you have a sequence upstream of these structural genes which is called as the leader sequence, and within the leader sequence this region is called as the attenuator sequence. We will discuss the function of the attenuator in the leader sequence later on. Now further upstream from the leader sequence you have an operator region, and upstream to the operator region you have the promoter region of the tryptophan operon where the RNA polymerase binds to initiate transcription. Further, many base pairs upstream you have the regulatory region designated as TRIPR. Now this TRIPR or the regulatory region is involved in the synthesis of the repressor protein which helps in tryptophan operon regulation. Now let us see the detailed steps of tryptophan operon regulation. So the first step, when tryptophan is present. So as I have already told you, trypoperon is involved in the biosynthesis of the amino acid tryptophan. So if tryptophan is present in the media, then what will happen? Bacteria will not need to synthesize tryptophan. So tryptophan operon needs to be switched off because tryptophan is already present. So what will happen? This regulatory gene with its own promoter region. So alright, this is the promoter region of the regulatory gene. So this TRIPR gene codes for this repressor protein, and this repressor is inactive form of the repressor. Only when this inactive repressor binds with tryptophan present in the media, this becomes the active form of the repressor. What happens next? Next, this active form of the repressor with the tryptophan binds to the operator region of the tryptophan operon. As a result, the RNA polymerase which is sitting in the promoter region is not able to proceed further, and transcription of structural gene does not happen. So the operon remains switched off. So this only happens when tryptophan is present in surplus quantities in the media. Now let us see the next case when tryptophan is not present in the media. So what will happen? The bacteria now synthesize tryptophan. Alright, because tryptophan is not present, the bacteria needs to synthesize tryptophan. So the regulatory gene, like previous case, will code for the repressor protein which is the inactive form, but because tryptophan is not present, this repressor cannot be activated. Okay, no tryptophan is present to activate this repressor. As a result, what happens? RNA polymerase, which was previously sitting in the promoter region, is free to proceed to the operator and thereafter free to proceed to the structural genes, resulting in the transcription and translation of the 5 structural genes, which brings about the production of the 5 intermediate enzymes involved in tryptophan biosynthesis, as well as a leader peptide is also produced from the leader sequence. Alright, so apart from the transcription and translation of the structural genes, the leader sequence is also transcribed and translated to produce the leader peptide. Alright, we will see the function of this leader peptide in the subsequent lecture. Now, if we zoom in into the leader region, so this is the leader region, and downstream to the leader region you have the 5 structural genes. Alright, so if you zoom in to the leader region, you will find it has an start codon designated as AUG. Then you have 2 tryptophan codons. UGG codons code for tryptophan, so you have 2 UGG codons, 2 tryptophan codons. Then you have sequence region 1, then 2, then 3 and 4. So there are 4 different sequence regions present in tryptophan leader sequence, and then you have a string of 6 U regions in the leader sequence at the 3' end. So this is the sequence of the mRNA which is transcribed from the leader region of the DNA. Alright, now there are 2 potential regions within the leader sequence which can form the stem loop or the hairpin structure. So the 1 and 2 region of the leader sequence can form a stem loop structure within themselves. Similarly, 3 and 4 region can also form a stem loop or hairpin structure within themselves. Alright, now 1 and 2, the function of forming this stem loop structure between 1 and 2 is that this formation of stem loop structure within 1 and 2 actually facilitates in an indirect manner the formation of stem loop structure within the region 3 and 4. Alright, and this stem loop structure formed within the region 3 and 4, this is the structure which actually brings about transcription termination in the case when tryptophan is present in abundance in the media. Alright, so this stem loop or the hairpin structure, this is the one responsible for bringing about transcription termination when tryptophan is present in abundance, and the first stem loop structure is actually helping to bring about the formation of stem loop structure between 3 and 4. Alright, so here transcription termination happens because of this stem loop structure. Now suppose 1 and 2, the 1 and 2 region do not form any stem loop structure, whereas 2 and 3 region form the stem loop or the hairpin structure. So this actually prevents the 3 region to form a stem loop or a hairpin structure with the 4 region. So this stem loop structure that is forming here, this similar kind of stem loop structure cannot form because 3 is already forming a stem loop structure with 2. So 3 is unable to form stem loop structure with 4. As a result, transcription termination can continue. So this happens when the tryptophan is not present in the media. Alright, so another factor that brings about transcription termination is that the position of the ribosome. If the ribosome sits on the region between 1 and 2, alright, so this prevents the formation of the stem loop structure within 2 and 3. As a result, what happens? 3 and 4 stem loop structure forms. So stem loop structure forms within the region 3 and 4, and just as I have already explained, this brings about the transcription termination. So if the ribosome sits before the region 1, alright, that is in the tryptophan codons, this position, if the ribosome sits here, alright, before the region 1, so what happens? This allows the formation of stem lobe or the hairpin structure within the region 2 and 3. As a result, because 3 is forming a stem lobe structure with 2, so 3 is not able to form a stem lobe structure with 4. As a result, just as explained before, transcription can easily continue. So this is the case of transcription termination. Now, why is it required? Sometimes the repressor protein which is produced and which goes and inactivates the tryptophan operon in the case where tryptophan is present in abundance in the media, that repressor protein along with the tryptophan is not that much strong enough to fully shut down the tryptophan operon. Then only the need arises for transcription termination by this stem loop structure.